Hello there, it's me again. I'm back with lecture number 14 in human biology, and the title of this lecture is DNA Structure and Replication. Now, we've talked about DNA replication in the past when we discussed the cell cycle. And when we did that, we broke everything into these phases, and there was interphase, and then there were all these mitosis phases, prophase, and metaphase, and telophase, and anaphase. But during interphase, we learned that is where the DNA is replicated, and it has to be replicated in order for the cell to divide and each cell to get a full complement of all of the DNA from all of the chromosomes. But we didn't talk about how that happens. It just kind of happened during the S phase in interphase. So today we're going to look a little bit more closely. We're going to look at it, drop down a little bit from the cellular scale to the molecular scale. And it turns out that at the molecular scale, DNA is super cool, and its structure has a lot to do with how it gets replicated. Here we have a funny with a scientist who is able to see the structure of DNA and as a result is trying to fix it. I don't know if that's possible or not, but it is the structure that makes that fix possible. And then there's another funny, which is a double strand, double helix of DNA saying to the other one, stop copying me. And it is the double helical structure which makes that copying possible. That's what we're talking. And on top of that, the structure of DNA is just super cool. So we like to look at it. We like to play with it. And I've done some of that myself. So let's talk about that structure and how it relates to replication. Okay, well, this is what we know so far, a little bit of review. DNA is in every cell, in every living thing, and in every biochemical process because DNA carries the instructions for making enzymes which control every biochemical process. So it's super important. But we didn't always know these things, and we certainly didn't always know the structure of DNA. So I've got a little bit of a history lesson here on what kinds of things led to us understanding that structure, which was a huge breakthrough in understanding how DNA functions. So we're going to start back with our friend Gregor Mendel and playing with the pea plants in the monastery in Austria. And in his notes and in his observations, he wrote that he believed that there must be some chemical that was carrying traits from one generation of plants to the next. So he was one of the first people to suggest that there was some type of material that must be transferred in the pollen from one parent plant to another when these pea plants reproduced. Later on, that's back in 1857. Later on, Friedrich Miescher found that if you looked at some sperm samples from salmon, pretty easy to collect, he found that he was able to isolate a substance that he called nuclein because it was from the nucleus of the cells in those sperm cells. And then a few years later, Richard Altman purified DNA from the chromosomal material. These were really just chemists who were trying to figure out how to separate the hereditary chemistry from all of the other chemistry. There was the idea that it was the chromosomes, because we had enough microscopy, it was the chromosomes that had that chemical information in them. That was the chromosomal theory of inheritance. And then came something really important. This is one of my favorite experiments in biology, from Frederick Griffith, who demonstrated transformation in bacteria. This is very cool. So there were bacteria that were dangerous and bacteria that were not dangerous. He used mice in his experiments. And it turns out that you could kill the ones that were dangerous. You could kill them, okay? And take a little bit of the fluid from where those bacteria were killed. And you could introduce that fluid over here to a place where the bacteria were not dangerous, and they became dangerous as a result, because somehow bacteria can take up the DNA of another bacterium and include its instructions in their genome. That's called bacterial transformation. And Frederick Griffith demonstrated that in 1928. That was a huge breakthrough in terms of understanding that this chemical material could be collected and could be isolated. And that's what we understand about DNA today. In 1936, whoops, I've got the wrong color here. In 1936, it was confirmed that DNA was the stuff. And then because we started taking DNA apart and trying to understand it, we saw that it was made of 4 very important types of parts. We've learned that DNA is made of nucleotides, okay? It is a nucleic acid made of nucleotides. Those are the monomers that the nucleic acid DNA is built out of. That polymer is built out of those monomers. There are 4 types of nucleotides that we need to understand. And Erwin Shagraf discovered that they paired up with each other in specific ways that made it possible to predict what kind of DNA we might have on one strand if we knew what the other strand looked like. It was in the 1950s when we first started to really scratch our heads about the structure of DNA, and I give credit to Rosalind Franklin, who was a X-ray crystallographer. She took pictures of molecules using X-rays, and she, in her notes, described that the DNA molecule was definitely helical. And then a little bit later, James Watson and Francis Crick kind of took over and took all the credit in terms of building a model that showed that DNA followed a double helix. And that's where we are today. But understanding that double helix is going to allow us to understand how it replicates and how the instructions are read to then be used to make those enzymes that control all the biochemical reactions. In fact, in their paper in 1953, James Watson and Francis Crick wrote, "It has not escaped our notice that the specific pairing we have postulated," actually it was Shagraf that postulated it, "the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material." Very cool, and it took a century to get us there, which is why I labeled this, "What a Difference a Century Makes." All right, so that's what we're going to look at today, the structure of DNA and how it relates to replication, and then next time, how we use it to record those instructions that make all of the enzymes. Okay, here's some more things that we already know because we learned them when we talked about organic macromolecules. DNA is a nucleic acid, and that is a polymer, and it is made of monomers that are called nucleotides, okay? The nucleotides are each made up of 3 parts, what I'm calling 3 subunits here. And we already learned about these, as I said. The first part is a sugar molecule, and that's called 5' deoxyribose. That's why DNA is short for deoxyribonucleic acid, okay? So deoxyribose is in there. That's this blue box here. That is the sugar that we see in that spot. The next part is a phosphate group, which is a phosphorus atom surrounded by oxygen and sometimes a hydrogen. But the ATE on the end of that refers to the oxygen. So that's a phosphate group. So it's a sugar, a phosphate group, and then the third part is the nitrogenous base. Nitrogenous is just a word that means that it contains nitrogen, okay? And we see lots of nitrogen in this structure. And the fact that it's a base is, it refers to the where it is in the molecule, but also that it's a little bit on the alkaline side, a little bit of less hydrogen in that area than there is in some of the other parts of the molecule. So 3 parts, the deoxyribose sugar, the phosphate group, and the nitrogenous base. And somehow we're going to take a whole bunch of these and put them in a big string in a polymer that allows us to record information about how to make enzymes. Whew. Okay, so here's a little new stuff, but I did mention this before. There are 4 different nitrogenous bases in DNA. So there are 4 different types of nucleotides that DNA is built from. 4 different types of nucleotides. But in each nucleotide, the sugar is always the same and the phosphate group is always the same. So it is the nitrogenous base that is different in each one of these nucleotides. There are 4, and they have names, and their names are adenine, which we abbreviate A, guanine, which we abbreviate G, cytosine, which we abbreviate C, and thymine, which we abbreviate T. A, G, C, and T. Those are the 4 nucleotides. Those letters really only refer to the nitrogenous base, but that's the only thing that's different from one nucleotide to the next. So we often refer to the whole structure as being A, G, C, or T. So one of the things that we notice when we look at these structures of these is that they come in 2 groups. C and T are molecules that we call pyrimidines. And the thing that makes them different is they only have 1 ring in the organic structure. G and A are a little bit bigger. They have 2 rings, and those are called purines. This is how I remember it. The shorter word refers to the bigger molecule. So A and G are purines, they have 2 rings, and C and T are pyrimidines, they have 1 ring. Shagraf, who did a bunch of work before Rosalind Franklin and Watson and Crick, discovered that when he broke down this material, he found that there was always the same amount of A and T in the compound, in the DNA, and there was always the same amount of G and C. And as a result, he postulated that A and T must somehow be tied to each other and associated with each other, and so must G and C. So we call these molecules, these pairs, complementary because they're always found together. So these 2, A and T, are complementary, and these 2, G and C, are complementary. And you'll notice that each one of those complementary pairs has 1 purine and 1 pyrimidine, and that's really important because we're always tying together a 2-ring molecule and a 1-ring molecule, so we're going to make a pair of them that are always the same size. That's really important in terms of the structure of this DNA. Wow, there's a lot going on here, so let me break it down for you a little bit. After we got to Rosalind Franklin and Watson and Crick, we had a structural model, a structural concept of DNA that described it as a double helix. That means there are 2 strands, 2 separate strands of these nucleotides that are twisted around each other, okay? And because they're twisted around each other, we had to think about the positionality of those nucleotides, and it turns out that the nitrogenous bases are toward the inside of that twisted pair, and the phosphates and sugars are toward the outside of that twisted pair. In fact, sometimes we refer to these bits on the outside as the phosphate sugar backbone, with the base pairs in the middle of this. If you can imagine this model that I have here just kind of twisted around, you'd see that this was the double helix. And we have a picture of it over here on the left with the 4 nitrogenous bases in the middle, and we can see those by color, and with the sugar phosphate backbone around the outside. And then if we zoom in on the molecular structures, we can see that here's the sugar, and here's the sugar, and here's the sugar, and here's the sugar, all right? The phosphate is between this sugar and this sugar, and this phosphate is between that one and the next sugar. And there's a phosphate here between these and here, so that's the backbone. And then in the middle, we've got T and A. Those are the 2 nucleotides that go together in this base pair, is what we call it. It's a nitrogenous base pair, always complementary. And we're showing that complementary nature by the shapes of these little colored bars because they kind of fit together. A and T fits together, G and C fit together. A doesn't fit that well with C, and T doesn't fit that well with G, so those are not complementary. And that's why we have them all lined up together like this. So double helix with the sugars and phosphates on the outside and the nitrogenous bases on the inside, and each strand has some directionality to it. It has 2 different ends. We refer to these ends as the 5' end and the 3' end. And you'll see the other strand has the 5' end up here and the 3' end down here. And that arrow suggests that the DNA is being created in the direction toward the 3' end. That's really important because that means that it's being built in 2 different directions. Okay, so that's going to come into play as we look at this. And that's what I mean when I say that they are anti-parallel structure, opposite directions, and also a little bit about the bonds that hold them together. The bonds that hold the base pairs together in the middle, those are pretty weak. Those can be broken pretty easily, and we can separate a couple of strands of DNA just by heating them up a little bit. But the bonds that hold the individual nucleotides together in a strand are a little bit stronger. And so it's easy to separate the strands without destroying the strands. Got that? That's really important. Let's move on. Okay, so now we know that a gene, which is the word that Gregor Mendel developed, is a sequence of nucleotides, a long strand of nucleotides that are in just the right order that they contain this information for making an enzyme. And it's on a single strand, so that there are actually 2 strands that could potentially contain these genes. So a gene is a long nucleotide sequence that's a set of instructions. How is that possible? How can we just put these 4 letters together? I mean, there's 26 letters in our alphabet, right? How can we just put 4 letters together to create instructions for making an enzyme? Well, it's because the strands are so long. It turns out that there are 3 billion base pairs in the human genome. That's just one copy, right? And we know that there are actually 2 copies in each double helix that we can talk about. 3 billion base pairs, which contains 30,000 genes and can produce 90,000 proteins. How big is 3 million? I thought maybe you'd like to have a kind of a reference for that. If I take my dictionary, which has 960 pages in it, I opened it up and looked at the back page, 960 pages. On each page there are about 50 lines, and each line there are about 80 letters. So just by doing some multiplying, the dictionary contains 3,840,000 letters. That's a lot. Well, if I took that and compared it to 3 billion letters, it would take 781 dictionaries in order to contain the information in just one copy of the human genome. And every cell in your body has 2 copies. So it's like a library right there in each cell. It's really amazing, and it has to be copied every time a cell divides, which we know in humans can happen about once every 24 hours. Woo! That's just mind-blowing. Big news. Okay, so now we have a picture of this structure of DNA as a double helix. And in this picture, we can see the base pairs with their colors and the phosphate sugar backbone on the outside. But that's a very, very long, thin strand. And what we've seen of DNA is these chromosomes during mitosis and meiosis, which are, I've described them as packages of DNA. So how does this DNA get packaged and unpackaged? Because in order to copy it, we need to unpackage it, then copy it, then package it back up. This is just getting more and more amazing. So I've mentioned to you that, that the stuff in a eukaryotic cell that we see, that we associate with DNA, is not just DNA. It is DNA plus some proteins, some structural proteins. And together, we refer to that stuff as chromatin. So one of the first things we learned about cells was that prokaryotic cells had DNA, but they did not have chromatin. There's no way to package that DNA up. Eukaryotic cells have chromatin, so the DNA plus the proteins are packaged up into chromosomes. What does that look like? Well, we just take this long flippy floppy strand, and then we wrap it around these pellets, which are called histones. That's a protein pellet. And then once we do that, we coil the resulting kind of ropey mess up into reoccurring loops. And then we take that looped up strand and we make it fold in and out of a structure that kind of looks like a flower. Sorry, that was a little too far. And as a result, we can stack them on top of each other. And in the end, it folds up into this chromosome, which if the DNA has been replicated, then it's a pair of sister chromatids attached at the centromere. So it has a lot to do with these proteins that the DNA is wrapped around and folded around and twisted around to make these chromosomes. And that's what I've got written up here. We need to unpackage it to read it and copy it. And then we package it by wrapping it around pellets, coiling it into chromatin fibers, and then condensing it down by folding it back and forth into this chromosome. Okay, so now let's learn some things about the replication of this molecule. So we're going to ignore the packaging and unpackaging. We're going to say we're in interphase, so we have unpackaged everything. We're not wrapped around the proteins. And we're going to take this flippy floppy strand and make a copy of it. So the way that we do that is that we take, let's see, here's a strand, and there's another strand right next to it. Let me grab a different color. And what we do is we create a bubble right in the middle, and that's what I've done here. We create a bubble so that the two strands are separate from each other right there. And as a result of separating them, we can then bring nucleotides into position and pair them up one at a time in order on each of these strands. And that's what I'm showing here. The nucleus of the cell is full of loose nucleotides. So once you've separated these, we can use enzymes to bring them into position and build another strand. So down below here, what we've got is that bubble that's formed, and we're always building our strands from the 5' end to the 3' end. It's always got to be in that direction. So on that strand, it's going, on the top strand, it's going to the left, and on the bottom strand, it's going to the right. And it always starts in the center, so that creates a bit of a problem for us, because if we start in the center, right here, and then we move to the left, then we can just keep building our new strand on and on and on and on like that. But if we start in the center and we want to build from 5' to 3' over here, we're going to start here and build a bit, and then we run into a dead end. So we have to come back here and build a bit, and then we run into a dead end. And then we come back here and we build a bit and we run into a dead end. So these two strands that we're making have different names. The one on the left here is called a leading strand. Okay, and the one on the right here is called a lagging strand. Unfortunately, they both start with the same letter, so that didn't work. So let me just highlight that here, leading and lagging strands. And that's also happening on the strand down below. Okay, so together, when we're forming these two new copies, we refer to this as semi-conservative replication. That means that we're actually making two copies at the same time, but one of the strands on each of our copies is a strand that originally existed in the first copy. All right, so we have the two gray strands in our original strand, and then in our two copies that we make, each of them is containing one of the gray strands. So semi-conservative because we conserve half of the original material that we had. That's why they call it semi-conservative. Whew, this is complicated. And I guess there's one other word here that I want to draw your attention to, and that is right here at this place where the strands are separating. That's called a replication fork. And you'll see those blue triangles there that are referred to as helicase. That is an enzyme that is able to separate those strands and start this process. It unwinds the helix structure and starts this process of copying. So it's all driven by enzymes. This is called semi-conservative DNA replication. All right, so it's a complicated process. It's complicated structurally, it's complicated biochemically. And as with most complicated biochemical processes that we've learned about. It involves enzymes, really important enzymes. I already mentioned one of them, and that is helicase, which is at the replication fork, unwinding the two places where the DNA strands come together. So it's working in two directions from that bubble. Just a few more, and there's 1, 2, 3, 4, 5, 6 of them here. This would make for an awesome multiple choice question. So helicase unwinds the strands. Gyrase breaks and holds the strands so that you don't get that twisty tension in it. If you grabbed some cord that was wound around in a helix and you tried to pull apart those two pieces of that cord, you'd get twists and turns in the rest of it because of the torsional tension of it. So gyrase is an enzyme that helps that become unwound. And the gyrase kind of sounds like something that's spinning, so that might remind you about that. There is an enzyme called primase, which begins the process. So it primes the engine, it gets it going, and it does involve RNA. There is an enzyme called DNA polymerase. This is basically the enzyme that brings new nucleotides into position and attaches them to each other one at a time. Polymerase just means it's making a polymer. It's an enzyme, ASE refers to enzyme, that makes a polymer. It's making the polymer of DNA. So it reads the template and inserts, if there's a T on the template is the strand that already exists. If there's a T there, DNA polymerase is going to move an A in there to be complementary with it. If there's a G there, it's going to move in a C. If there's an A there, it's going to move in a T. It's always going to bring the right nucleotide into position. That's DNA polymerase. The next is ligase, and ligase comes into play with those lagging strands that keep running into the dead end. Those are called Okazaki fragments. I think somebody in Asia clearly described them the first time. And basically, once you run into a dead end and you start behind it a little bit, you actually have to seal all of those fragments together. And so we have a special enzyme for sealing up all those pieces of fragments. And then the last enzyme is called telomerase. And there's a funny thing that happens when we get to the end of a long strand at the far end of the arm of a chromosome, where we don't quite have enough for the other enzymes to work properly. And so we end up losing a little bit of our ability to make a copy at the very end. And we have a special enzyme that allows us to add that little bit onto the end. It's only for that end piece. So it says that it finishes synthesis at the end of the chromosome on the lagging strand. Helicase, gyrase, primase, DNA polymerase, ligase, and telomerase. So that's great, but let's not forget that in addition to making a copy of the DNA, it also has to be checked. Remember that in interphase, there was the S part, which was synthesizing DNA, but there was also G1 and G2, which were checking it. And the nice thing is that it involves the same enzyme. DNA polymerase is an enzyme that can proofread the DNA to make sure that all of the nucleotides are in the right place. So this kind of orangish ball here is supposed to be DNA polymerase, and it's moving along the two strands, and it comes to this point where, oh my goodness, we've got a G and an A right across from each other. That's not right. They don't go together. They're not complementary. We need to fix that, and there's a few different ways to fix it. And over here, well, we're showing one of them. It's called mismatch repair. Over here, we've got a G and a T that are across from each other, and that's not right, so we need to fix that. This is the fixed version of it, where we've replaced it. And then also sometimes when it's a really bad mistake that we might make, we use a process called nucleotide excision, where we can cut a big chunk of a strand out from one end to the other and then replace the nucleotides in the middle. Anyway, it has to be checked in order for these repair processes to take place. And DNA, because it's constantly being replicated, is also constantly being checked and repaired. Remember that scientist that was nailing some boards on the DNA at the beginning? DNA repair is part of a natural function of cellular function, cellular replication, but also these things come into play when scientists want to play around with the structure of DNA. They can separate strands, they can cut out pieces, they can put new pieces in. It brings us face to face with the power that comes with understanding how DNA functions. And that's scary sometimes. Amazing, but scary. And just as I'm wrapping up here, I thought you might like to see that because the structure of DNA is so important to the function of living cells and living organisms, and because it's so cool, we love to build it. We build it all over the place. And all of these companies are making these models so that students can understand this structure. You can see them from in all kinds of different ways fitting together. You can see the nucleotides, the phosphate sugar backbone. You can see that they can get really fancy and complicated, including all of the individual atoms, and that the students building them are very fascinated and excited to be putting this DNA together. You can see that sometimes they're so excited that they don't worry about getting something fancy. We've got macaroni, we've got gumdrops, balloons, and beads, and pom-poms, and there's a person there on the refrigerator. You know what? If you're hanging it on your refrigerator, you know you're proud of it. So some great students getting into the action here in terms of building DNA. And some others, we have, look, there's somebody who's doing crochet. We've got Legos, we've got licorice and marshmallows, snacks, soda cans, all kinds of stuff. And then artists also get into the game in terms of taking advantage of the excitement that people have. And there are sculptures all over the world that focus on this structure. And these are some of my favorites. This one here is like a spiral staircase, and you can walk to the top of it. This one here is carved out of solid wood. That was a lot of work. Here you see one that's at the Genetics Institute in Ireland where I visited. Here's one that's made of money, and DNA is becoming more associated with that. This is my favorite right here. This one is made out of shopping carts. And that's just cool. How could that not be cool? Anyway, lots of representations of this structure. Okay, wrapping it up, and this is a preview of what we'll be talking about next time. Today we've been talking about DNA and how it is replicated and how it is checked for mistakes and fixed, and we get ready for mitosis or meiosis, making copies of the chromosomes. But there's another nucleotide that's really important, and that is called RNA. There's only one letter difference, and you know that letter D refers to the sugar, deoxyribose. So the letter R in RNA also refers to the sugar, and that's just ribose. And the reason it's different is because RNA is generally single-stranded, not double-stranded, but it is also made out of nucleotides. And the reason it plays an important role is this is how we take that information in DNA and use it as instructions for building enzymes. We take the DNA and we copy it into RNA, and that's called transcription. And then it's on one strand. And then the RNA exits the nucleus and finds itself a ribosome, which is where proteins get synthesized. And that's called translation. And this whole mess of bob together is referred to as the central dogma of molecular genetics. It is so important, so foundational. They're calling it the central dogma. What we'll learn right away when we start looking at RNA is that it's not quite the same as DNA because it's not double-stranded, but because there's one nucleotide that's different. It doesn't have thymine in it. It has something called uracil in that spot. And so next time we're going to learn about how RNA is involved in this process and how we get an opportunity to take our copied DNA and read it and make some proteins out of it. Stay safe, be good. I'll see you in class.